Hey guys, happy Wednesday. It's the middle of October. We're moving, we're grooving. For some of you, you're already done with your first quarter of school. If you're like us in the South, we started in like the middle of August, which is amazing to think we were already 25% of the way done. For those of you who still have a little bit longer, you're getting there. You're real close. I promise. Thank you for putting up with my slight delay. I have the world's oldest not old computer, so sometimes it takes a minute longer than I think to load. So sorry about that. Here's my regular tech check. Can you hear me? Please tell me if you can hear me, because I'm paranoid. Yay, thank you, Sandra. Hi, Sandra. Good to see you again. Hi, Omi-san. Hi, I'm so glad you're here. I'm creeping on who's here now. Jody, Nayeli, Dolores. Yay, I'm so glad you're all here. Okay, so we're gonna have a good time. We're gonna talk about mechanisms of membrane transport. Just so y'all know, this is gonna be a little bitty bit of a review session. So a lot of this stuff, I'm gonna be perfectly honest, I couldn't think of any better way to explain it than I have previously. So a lot of these slides, if you've seen this before, like Sandra, you've been here so far for everything, they're gonna be repetitive. But the reason that I assume College Board has this standard is to really, really drill in. This is why this is important. And this is going to be something that's critical throughout your class. So I can tell you right now, just from my experience as an AP biology teacher, that you're going to need membrane transport throughout everything. You're going to need it, especially in cellular communication, which is our next big unit after photosynthesis and cellular respiration, but you're gonna need a lot in there too. So that's why we're doing a lot of review on this. I've got review on all the topics, and I think some new stuff, because I think we're a little bit out of order. I don't think you guys have done tonicity yet, at least not through Fiveable. So we're gonna talk a little bit, just little bit preview on tonicity, and then we are also going, sorry, my watch dings a lot, and then we are also going to do a practice FRQ, which I know is what people really like, because then you're like, yes, preparation, because if you're anything like my kids, my 95 darling angels, FRQs are fun, so, but you want practice at them, so we're gonna do that. Okay, so let's go ahead and get started. So regular plug, please follow Think Fiveable, or follow me, Jessica Nadsam, your guide on this journey today, to see some cool stuff that we do and what we are all about. We would love to see it, see you there. If you do some cool stuff, feel free to tag me, because my kids are always like, Miss Nadsam, we want more labs, we want more cool stuff. And I'm like, hi kid, help me find it. So if you know of anything cool, feel free to share it with me. If you want your classes to have some cool stuff, feel free to creep on me on the Twitter, because I have so many things I put on the internet, because I'm a millennial, and if it's not on the internet, it's not real, obviously. Okay, I just had a lot of caffeine and a lot of sugar, so I apologize for the level of energy I have at this point. If you're on the West Coast, this may be a normal level, because I guess it's like 5 o'clock for y'all right now, but I know those of you on the East Coast, it's like 9 o'clock, and so, well, maybe you guys don't go to bed as early as me, but my bedtime's in approximately 27 minutes, so that's how it works. Okay, so going back to our agenda today, we just did our intro. Great job, everybody. We're gonna review passive transport, we're gonna review active transport, we're gonna review bulk transport, we're gonna go boom, 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 boom through these, because again, this is a review, but we do want to emphasize how important those are and how much you're going to need them when you get to later units. Because if you don't understand these, you can get through later units and it'll be fine, but your head will hurt significantly more than if you just knew it the first time over. So we're also going to talk about osmosis and tonicity. We've kind of touched at these when we talked about water and the properties of water before, specifically if we touched into water potential. But we want to talk about them just a little bit more. Notice I'm saying a little bit a lot today, which means there's a lot of little things. And then obviously the part that everyone really, really likes, AP exam review, because everyone wants a sneak peek at some of that stuff that may show up on your test. So here we go. All right, quick review here. So these things, phospholipids. We have talked about these before. Who can tell me which part of the phospholipid is the polar part? Which part of the phospholipid is the polar part? The head. Very good, Sandra. What molecule is it in there that's making it polar? What molecule is making the head polar? Or element, if you want to list the element. Here, I'm going to blow this up for y'all, make it a little easier for y'all to see. So in this head, there's some specific atom or element complex thingy that's making this polar. What is that? Oxygen. Very good, Sandra. Remember, guys, oxygen is highly electronegative. It is super greedy. It did not have siblings, apparently, because it never learned to share. And so oxygen is an electron hoarder, and that's going to make it super, super polar because it's got a very unequal charge distribution. And then the tails, on the other hand, are going to be nonpolar. So you would think these two wouldn't go together, but they have some just magic chemistry that makes them work. And it's great because they are amphipathic, and that's what's going to make them form these bilayer sheets that are in all your cells and make your membranes all wiggly, and we like that. So we're happy with them getting along. Any questions about phospholipids? Take 3, take 2, take 1. Okay, I'm going to call it good. Let's talk about the plasma membrane a little bit more. So again, this is made up of phospholipids. And again, guys, I'm sorry if you've been with me before. These slides are a little repetitive, but to be perfectly frank with you, I spent a lot of time searching for these GIFs. And so if I picked them, it's because I think they're perfect. So the review is not going to be that original, but I'm going to try to come up with some extra special questions for you. So speaking of which, we say that this membrane is flexible. Why is this membrane flexible? What is holding these phospholipids together? Why is that membrane flexible? Why is the membrane flexible? Why isn't it solid or crunchy? Because we know it's flexible because I can do all sorts of stuff to my arm and it doesn't break. I'll give y'all 3, 2, 1. All right, so let's talk about the flexibility of this membrane because as you remember, we call this thing a fluid mosaic. We're going to cover that more in our review in a moment. But the reason this thing is so flexible, there are very weak attractive forces that are actually holding these things together because the tails are holding each other together, the heads are holding each other together. And so what you see is these things actually kind of slide over each other. And so it makes it very, very flexible. We're going to see a GIF of that. But what's another word for weak attractive forces? This one I know someone here should know. What's another word for weak attractive forces? Weak attractive forces. Come on, I know someone knows this one. Your teacher talked about it a lot when you covered water. Hint, hint, nudge, nudge. Another word for weak attractive forces. Is it a type of bond? Yes. It's a certain type of bond. What type of bond is it? It's weak, but it enables a lot of cool stuff. Type of bond. It's one of the first ones you would have learned about in biology. Water is really good at it. Hydrogen! Yay, Omnisan, good job. So it's a hydrogen bond. So these things are held together, very weak attractive forces, hydrogen bonds, and that's going to let them slide to the left, slide to the right. Dun, 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 dun. That's what they do. And that's what makes your phospholipids flexible, because if I have some skin, here's my arm. Here, I'm going to make this big so you can see my arm. Or here, better yet, here's my face. My face is flexible. Don't touch your face like this. It's not good for it. I'm doing this for science and for your education. But I can move the skin on my face and it doesn't break. Even on skin that's more tense, like your hand, sorry if this is bothering anyone, but I can flex my skin and it's not breaking it. It's just fine. The reason is because you have those flexible phospholipid bilayers. If those were solidly stacked together, every time I did this, they would crumble and my face would break. And I don't know about y'all, I like my face the way it is. Hopefully you like my face the way it is. So this is going to be the skin of the cell. It's going to be the barrier that separates everything. So I don't know if it's on this slide or the next slide. Now we'll come back to this. What kind of stuff is able to pass through that plasma membrane? What kind of stuff passes through the membrane easily? No problem, easy peasy. Small and uncharged. Very good, Sandra. So you're going to want very small particles. And you're going to want non-polar particles. Anything that has a charge, anything that's polar, it's going to do this thing where it just smacks into that plasma membrane. And the polar heads are cool with it. They're like, yeah, come on, party. But the non-polar tails, as soon as something polar hits those non-polar tails, they're like, can't come in. You can't sit with us. That's what they say because they can talk. And so then, as you can see in here, we've actually got stuff that's bouncing off for a different reason. But there's other stuff bouncing all over the place in here, and it's not getting through because it does have that polar charge. Meanwhile, it can also be too large. So like these guys, I'm going to assume these are proteins. They're huge, and they're just kind of bouncing into the membrane and not able to pass through because they are too big. So that's going to be very important. One more thing I want to emphasize here, the fluid mosaic model. We call it fluid because it's flexible. Why do we call it a mosaic? Why do we say the cell membrane is a mosaic? Tell me things. Why do we say the cell membrane is a mosaic? Why? So many different parts all together, very good. So what are some examples of those parts? What are some of those parts that are on there? The many proteins, very good. Very good. What else is in that cell membrane? I'm going to make it big again so y'all can see. They're not labeled, but phospholipid bilayer, embedded proteins. There's one other thing. It's really important for insulation. Baby seals need this a lot because it's cold where baby seals are. Cholesterol, fat, very good. So you're going to find cholesterol. Remember, fat, is it polar or non-polar if it's fat? Polar or non-polar if it's fat? Non-polar, very good. Very good, Avery, and very good, Sandra. So non-polar. So as a result, that cholesterol, which is a fat, is also going to be non-polar. So it's going to mix with the tails of those phospholipids. So you're going to find it right in here. And that's going to give it a certain level of flexibility, but also structure and insulation. So if it gets too cold, it doesn't literally freeze. That's what prevents your body from actually freezing, it turns out. So good job, y'all. All right. So let's talk a little bit about how stuff is actually going to get in and out of this membrane. So got a couple things going on here. Got a lot of molecules in this picture. One thing I really want to stress is, again, it's all about the polarity. If these things are polar, they will not pass through the membrane. They may pass through something else, but they will not pass through the membrane if they're polar. If they are big, they may get one through because it's just gonna red rover its way. But that's a whole different type of transport we're gonna get into in a moment. Reality, you're gonna need a protein for that sort of thing. But also, one thing I really wanna talk about here is what drives the direction that those molecules move? Do these molecules have like a sentient brain that says, "Ooh, must go inside the cell, because if it don't go inside the cell will dehydrate and then it will shrivel up and it will be sad." Cells aren't sentient. They're just a bunch of atoms and carbs and proteins. Good, Avery, the concentration gradient. Yes, solving it before I could even ask the specific question. Very good. So this is going to be not a sentient cell thing. You must put water in the cell. It's all about diffusion. So the way that I do this with my kids is, and they hate me for it for like a day and then they get over it. Oh, it's okay, Avery. I appreciate it. It was awesome. But so what I do with my kids is I go in my classroom and there's like 20 of them. And you know how in a classroom, there's like those linoleum tiles that are like 12 inches by 12 inches. So it's like a square foot. So I make every kid stand in their own tile. Okay, cool. You fit 2 feet in 1 square foot. That's fine. But then I make them put all the tiles next to each other, like checkerboard style. And so these kids are there's 20 kids in 20 feet by 20 feet. And so the kids are highly concentrated and they don't like it and they want to leave. And I'm like, I get it, but I got to make a point. And so then I say, you are currently highly concentrated. If I was to suddenly give you 40 square feet or 40 squares, what would you do? And they were like, we would spread out. Ding, 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 ding. Things like to move from high concentration to low concentration. The other way I like to think of it is if you go hiking, or if you've ever taken your dog on a walk in a hilly neighborhood even, if you've ever been on a walk in a hilly place, if there's a hill at your school, whatever it is, what's easier? Climbing up the hill or walking down the hill? What's easier, climbing up or walking down? Which one is easier? Going down. Thank you, Sandra. Yeah. If you're riding your bike, you don't want to pedal up a hill. You want to coast on down. Same way with the concentration gradient. You want to start at the top of that hill and then you want to say, see ya, and just whoop, on down. That's easier. It doesn't require any energy. And these molecules are going to be exactly the same. So when you start out with all this glucose over here, glucose is this red one. So you've got like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 glucose on the outside of the cell. You've got 3 glucose on the inside of the cell and 1 that's on its way. That glucose is naturally not going to want to all be next to each other. No one wants to be that close to everyone all the time. If they do, good for them. But everyone needs a break every now and then. So that glucose is trying to spread out down to that lower concentration. That's going to be diffusion. That's going to be along the concentration gradient. Dun, dun, dun. Any questions about the concentration gradient? This is an abstract one, so if something pops in your head, please tell me. I really want to help you here. What questions do we have about the concentration gradient? 5, 4, 3, 2, I can't count, 1. Okay, I'm going to move on from the concentration gradient. It's done. We can never go back. Okay. So passive transport is going to be that illustration of the concentration gradient. So we have 3 stages. One where all of this, what's going on? Hang on, my screen's being, there it goes. Okay, sorry, my screen was being weird. But so we've got all these little hexagons. I'm going to say since it's a hexagon that it's a glucose. That's just the typical shape in biology that we draw glucose as. So I'm going to say this is sugar on the outside of the cell, the extracellular fluid. And let's say these are my cells. And I just drank a milkshake because I knew I would need some energy for you guys, to be perfectly frank. So I had my milkshake. I've got all this sugar. It's currently sitting in my belly, and I'm just waiting for it to process. But it's got to get into my cells to deliver that sugar. So this is the current state outside my cells, extracellular fluid. And again, the sugar does not like all being so close to each other. So it's going to slowly float across the membrane until it is in equilibrium on this side. It's all on the same side. What is wrong with this diagram, though, if that's glucose? If it's glucose crossing the membrane, what's wrong with this diagram? That's glucose. What's wrong with this diagram? What's missing? Think, think, think. It should have something that it should be able to shoot. Very good, Sandra and Avery. It needs a protein channel. It needs its own passageway. Why can't glucose just pass through the membrane on its own? It's too big. Very good. Glucose is too big to just pass through a membrane. It doesn't fit. And so as a result, it needs that protein channel. And what do we call it when we use a protein channel to do diffusion? What do we call that? Facilitated diffusion. Very good, Avery. So that's when we get to this facilitated diffusion. And we've got stuff that's going in and going out and all this. Does it require any energy to do facilitated diffusion? Does it require any energy? Nope. Why not? Why doesn't it require energy? Concentration gradient. Very good. So since we're still going from that high to that low, we are riding our bicycle down the hill. It doesn't require any energy. Very, very good. So the only difference with facilitated diffusion is it is going to have those proteins that open and shut, or they may just be a tunnel. Either way, stuff is now able to get through and it doesn't have any issues. Any questions about facilitated diffusion or about diffusion in general or passive transport? Speak now or hold your peace until you have a question later, because I'm not going to tell you forever. Okay, let's talk about some active transport. What's the difference between passive transport and active transport? Tell me your secrets. Tell me what you know. I know you know it. Active transport requires energy or ATP. Very, very good. Sandra, good addition because it goes from low to high. So this is like when I push my kids in a corner in 20 square feet with 20 kids where I say, okay, you all have your own box, but you must stand in your box and your box must be next to someone else's box. And then my kids look at me like I just told them there's going to be a pop quiz on something they haven't even learned, which I would never do. I'm not that mean. But I would make them stand in a corner if it was to accurately illustrate diffusion. So this is like when you're pushing a bunch of things closer and closer and closer and closer together. You are putting them in a state of higher concentration. It is akin to riding your bike up a 90-degree angle. It is not a pleasant experience, but it's possible if you expend a lot of energy. So you've got to pay ATP for this. I'm not going to go into the specifics of ATP because that is more of a like photosynthesis and cellular respiration unit, but think of it as a coin. And every time you want to put one over, you got to put a coin in the slot. It's like whenever you play that stupid game at the grocery store where you put a quarter in and you get a chance to like use the claw and it can come down and like grab a stuffed animal. It's like that. Every time you want to play the game, you got to put a quarter in the slot. Every time you want to move something across the membrane from low to high, you got to put an ATP in the membrane. That's all you really need to know right now. There may be more later, but we'll get to it. Any questions about active transport? 4, 3, 2, 1. All right, here we go. Go team. All right, so this is an active transport example that I understand has already been talked about in some other streams. I know I've looked at it before, so I'm not going to go super in depth into this one. But can I go over co-transport? Ooh. I'm going to be perfectly honest. I haven't talked about co-transport in a while, Omi-san. Give me just a sec to refresh my memory. So co-transport, because I've got my notes on my Google Drive. One sec. Okay, so co-transport's a funny one, and I'm going to be perfectly honest with you. I have not seen it in the AP biology. Yes, and Avery, we are going to go over bulk transport, so good question. But Omi-san, I'm going to be perfectly honest. I have not seen co-transport in the AP biology syllabus. That does not mean your teacher is not teaching it. They may be doing it additionally because it is valuable, but I haven't seen it in the AP bio syllabus, so I haven't taught it in a while. Co-transport's funny though, because I'm going to make my face bigger for a sec. So co-transport is kind of funny. So it's like, you got your cell membrane. You don't see that? There's your cell membrane. Pretend it's a cell membrane. And you've got a protein. And the protein goes and lets stuff in and it lets stuff out. But this protein's interesting because it lets some stuff in. That's not a straight line, bear with me. And it also sends some stuff out. So you would think of this as being an example kind of like a sodium-potassium pump, because sodium-potassium pumps kind of do the same thing. They put something in, they put something out. Co-transport is going to be a little different because co-transport is going to have one that is going with the concentration gradient, but the other molecule is going to be going against the concentration gradient. So it's doing two different molecules, and one is going to cost energy, and the other one is just going to spit it out because it's going to go with diffusion, or it's going to go with facilitated diffusion. It's going to be going along its concentration gradient. Does that kind of help, Omi-san? I'm sorry, I didn't have a slide prepared. I was a little off guard, but I hope I helped. Please validate me. Yes, okay. And if you have any more questions about it, please let me know. I'm happy to help. I just had to make sure I didn't mess up the explanation because I'm human and I do that every now and then. Okay, Avery, bulk transport. We're gonna get to that slide because I came prepared. I do have a slide for that one in just a second. Great. So sodium-potassium pump, I'm going to make this big again because you don't need to see my face. You need to see proteins. So you've got your cell membrane. You got some potassium. You got some sodium. These are going to be going active transport. It is going to require energy because it's going against the gradient on both sides. And so the protein works kind of like co-transport for both of those molecules, but both of those are going to be active transport. So I know that one's been talked about a lot, so I don't want to spend a lot of time on it. Does anyone have any questions about the sodium-potassium pump, or do you feel like you're pretty good? Okay, I'm going to go ahead and move on. But if you think of something, please feel free to let me know. Bulk transport! I love this one because I have this cute little GIF and you can see this cell and it's eating another cell. I mean, come on, y'all. Look at this guy. He is chowing down on another cell. He's got his big cell mouth going, ah! And he just says, chomp. That's bulk transport. It's when the cell membrane, so you can kind of see it over here with endocytosis, because this is an example of endocytosis. So this foreign particle, it could be another cell, it could be some giant food molecule, it could be a lot of different things. It can be anything that's big, basically. And so what it's going to do, it's basically just going to punch into the cell membrane, or the cell membrane is going to chase it, or the cell is going to chase it and chomp it. And it's going to fold the cell membrane around it into this vesicle. You can see one right here. And then it's actually going to pinch in and bring that vesicle into the cell. And then it may release it, do with it what it pleases, eat it, digest it. If it's something that may harm the cell, it may be digesting it so it can make sure it doesn't harm the cell. So that's that on endocytosis. Endo, like enter, as in to come into the cell. And then on the other side, you've got exo, as in exit. So you're literally just doing the reverse. It's got these little things in here. They kind of look like bugs to me, so I'm going to call them bugs. So we got these bugs, and they need to get them out of the cell. And so what they do is they form this vesicle around it, made of the cell membrane once again, and then they just spit them out. So picture this GIF just playing in reverse. So bulk transport is pretty simple. Just that exocytosis and that endocytosis. Does anyone have any questions about that? Did that help, Avery? Phagocytosis and pinocytosis. Okay, so very good, Audrey. So phago comes from the Latin word phago, meaning to eat. And so that's literally what this is. This cell is eating another cell. And so phagocytosis is usually going to be an example of endocytosis when it's eating some large amount of food. Pinocytosis, pino meaning to drink. So with your pinky out because you're fancy, you're going. But pinocytosis is when it's basically doing this, but it's doing it around water. And so it's like taking a big gulp of water. The best way I can describe phagocytosis, if you've ever seen those whales that just swim around with their mouth open, like, and that's how they eat the little fish, that's the best example of phagocytosis I can give you. Does that make sense? Yes? No? Awesome. Happy to help, Avery and Audrey. Does anyone else have any other questions about bulk transport? Okay, I'm gonna go ahead and move it on to a summary page. Ooh, I love summary pages because it's just an easier way to study. You can see everything right there. Guys, my shirt matches the summary page. Sorry, I've been up since 5 a.m., so I'm a little distracted. But I didn't plan that, but I feel pretty cool being able to do that. So, okay. So the main things you need to get out of this: what takes energy, what uses the selective membrane, which ones use proteins, and which one follow the gradient. So active and passive transport obviously are going to be opposites. Active requires energy, passive does not. Bulk transport also is not going to require energy because all it's doing is going. Selective, active transport and passive transport are both going to be very selective. Only certain stuff can pass through depending on its polarity and if it has a protein. Very, very picky. I said no for bulk transport because you're not passing through the membrane. It doesn't require that non-polarity or that smaller size. But I do imagine cells don't just do bulk transport on anything that pokes them. So I do believe they are slightly selective. But when I say selective, I'm thinking of that non-polarity and that size. And that's not going to matter with bulk transport. Proteins. Active transport is going to use proteins. Passive transport may use proteins. It may use diffusion. It may use facilitated diffusion. So it's a yes and a no. Bulk transport obviously is not going to use proteins because they just, again, go daddy shark style. And then the gradient, active transport is going to go against. Passive transport is going to go along. Bulk transport doesn't care. It's going grandma shark style. Gotcha. Any questions about this summary? It's very short. Okay, to confirm. Channel proteins are for facilitated diffusion with no energy, and carrier proteins are for active transport with ATP. Okay, so Avery, ooh, you guys have such good questions. So I'm going to be perfectly honest. I call channel and carrier proteins the same thing in my class. I don't have a difference for them at this level of biology. They're both proteins. I don't use a specific channel versus carrier for them, but I don't want to mess you up in case your teacher does. So Avery, I think what you're saying, channel proteins are for facilitated diffusion, carrier proteins are for active transport. I think that's a good way to keep it in your head. I personally don't have a difference with them, but that may just be a difference between my teaching and your teacher's teaching. Does that make sense? Just how in depth we go with the vocabulary. Does that make sense? Okay, awesome. Because I don't want your teacher emailing me later being like, hey, why'd you mess her up? I'm going to be like, I didn't do it. And she's going to be like, I have video evidence of this. Okay. So tonicity and homeostasis. Sandra, you've been on a lot of these things. Has tonicity and homeostasis been covered? I don't think it has. I think it's scheduled for this Sunday. Have you guys talked about tonicity and homeostasis in class? Yes? In class. Okay. Not yet. Okay. So test is tomorrow. Hopefully. I can see. Very good for you being here. So I'm not going to go super in depth in this because there is a whole hour-long spiel on it. So I'm not going to go super in depth in it. But I'm going to touch on it because it does play into this. If you remember with water, water is very specific in where it moves, and that has to do with its hydrogen bonds and adhesion, cohesion, all those things, being the universal solute. But water is basically going to move to dissolve things as best it can until concentrations are equal. It doesn't like one side to be more concentrated than the other, whether that's a cell in a beaker, which is the big example you're going to keep seeing for the next 3 days if you haven't had this yet, or whether it's a bunch of solute just in a jar. So this is going to have to, this is the ability of a extracellular solution to make water move in or out of a cell. So you can see here, I've got animal cells and plant cells. I've got cells that are literally exploding, cells that are happy, and cells that are shrinking. This all has to do with the type of solution you put in them. So again, I'm not gonna go super in depth with this one. The main thing I'm gonna say, and I'm gonna come over here to do it. So like right here, you can see I've got a low concentration on one side and a high concentration on the other. And you can also see these molecules are too big to pass through this semi-permeable membrane. The molecules can't move. They're stuck there, but the water can move. And so the water, we can see here, it moves all to this side, even though gravity says it should be falling equally, but it all moves over here because that makes this side less concentrated. So this is going to be less about the movement of molecules and way more about the movement of water. That's why we call it osmoregulation, like osmosis, osmoregulation. It's the maintenance of constant osmotic pressure in the fluids of an organism by the control of water and salt concentrations. Okay, that's a mouthful. The main thing that you need to know is water is always going to move to make things less concentrated. Water does not want things to be different concentrations. It wants them to be equal concentrations because it's all about balance in yoga and Zen. I want to ask any questions just because I know some of you have already covered it. So like Avery, you said you've got a test tomorrow. Do you have any questions on this that I can help you with before your test? And any others who do have questions, please feel free to ask them. I just know I did not go very in depth, and that in-depthness is coming, just so you know. Okay, I'm going to go ahead and move on. But if anyone thinks of anything, please feel free to do a smoke signal and let's talk about it. All right, last thing for the night, y'all. Hey, here we go. So I'm sorry this font is kind of tiny, but I wanted to fit it all on a single slide, and it's just what happened. So it says here that the following experiment was designed to test whether different concentration gradients affect the rate of diffusion. Concentration gradient, remember that has to do with concentration of solutes, high to low, In this experiment, 4 solutions, 0% NaCl, 1% NaCl, 5% NaCl, and 10% NaCl were tested under identical conditions. 15 mils of 0% NaCl were put into a bag formed of dialysis tubing that is permeable to sodium chloride and water. The same was done for each NaCl solution. So all 4 solutions have their own little baggie with 15 mils. Each bag is submerged and merged in a separate beaker containing 300 mils of distilled water. The concentration of NaCl in milligrams per liter in the water outside the bag was measured at 40-second intervals. The results from the 5% bag are shown in the table. So this is our 5% bag, and this is how much water was outside the bag. So I'm going to make this small for a moment because I want to draw a picture. So here's how I think of it. You got a beaker. Beaker's full of water. In that beaker, you have this little baggie filled with NaCl. So you got a baggie filled with NaCl in this thing of water. Can the NaCl cross the membrane? Can the NaCl cross the membrane? Read the directions here. Can the NaCl cross the membrane? Yes, very good, because it says here, a bag that is permeable to sodium chloride and water. And so we see here outside the bag, the longer that time goes, the amount of sodium chloride in the water increases. So what's happening, let me make this big again. The sodium chloride is all leaving the bag. It must be. Okay. So part A says here, on the axes provided, graph the data for the 5% NaCl solution. Y'all, I'm sorry, there's not really a way for us to do this, but I want to go over what that's going to look like. So you're going to have to use this information and you have to graph it. Here's the critical thing you must know about graphing when you do a free response. It's almost always going to be 3 points. First point is just going to be for graphing it. Second point is going to be for scaling it properly. Third point is going to be for labeling your axes with units. So seconds and milligrams per liter. Guys, I was an AP reader. I graded 5 zillion AP tests last summer. I know how this works. And there were so many kids I couldn't give a grade because they didn't put milligrams per liter and it crushed my soul. So keep that in mind. So I'm going to go ahead and show here. So for part A, you can get 3 points maximum. Correct orientation of the dependent variable on the y-axis and the independent variable on the x-axis. Correct axes labels and correct plotting of all the data points. So what this is going to mean, I'm going to draw you another picture because I'm nice. You're going to get a graph. It's going to be a box. It's going to be prettier than this, but I'm going to hurry. You're going to have your graph. What that means is you've got to do units like 1, 2, 3, 4, 5, and you've got to label down on the bottom. I know this is backwards. I'm sorry. That's not why I'm bothering. That's why I'm not bothering to draw the real thing. So you got to label it. Then you got to do some units like 1, 2, 3, 4, 5 up here, and you've got to label with units on the side. And then you've got to draw what's actually happening. Is the line going up? Is the line going down? What's it doing? That's how you get 3 points. Whenever you do a free response question, I always encourage my kids to do the graphing first because it's the most bang for your buck in terms of points. It's 3 points just for drawing a graph. And most of you, if you've been in AP for a while, you've been drawing graphs since like 6th grade. So just draw the graph and make sure you remember units. Okay, part B of this, I'm going to come back into a bigger screen because I'm going to go back for a second. So part B is going to ask you, using the same set of axes, draw and label 3 additional lines representing the results you would predict for 0% NaCl, 1% NaCl, and 10% NaCl. Explain. So this is another good one because you just draw and label it. So you're literally just going to take your graph and draw some extra lines and label, this is 0, this is 1, this is 10. What is that actually going to look like? So again, this is a 4-point question. So just predicting where it goes, 0% is going to have a flat line because if there's no NaCl, it can't move out. 1% is going to be somewhere below 5% because there's not as much. 10% is going to be somewhere above 5% because it's going to have more. Typically, when it's asking you to make a prediction like this, you don't have to get specific. There's not like a magic, oh, you must draw the line like this and then turn left it there. You don't have to do that because you're not expected to be able to get it that perfectly. But you got to have it in the right ballpark and you don't want to go too far left field. So that's going to be your example there. But then you have to explain it. You'll get a point, 1 point if you get all 3 right. You don't get a point if you only get 1 of them right. And they don't give you half points on this. But the explanation, for every explanation that you come up with for the 0%, the 1%, and the 10%, that's a point. So, points, points, points. Any questions about this free response prompt so far? Okay, I'm going to go on to part C. So part C is going to say farmlands located near coastal regions are being threatened by encroaching seawater seeping into the soil. In terms of water movement into or out of plant cells, explain why seawater could cause decreased crop production. Include a discussion of water potential in your answer. So again, we haven't talked much about tonicity yet. We haven't talked much about that water potential. That's something that is coming, but I wanted to go ahead and give y'all a taste of this. Let's talk about seawater because they'd love to use it in AP questions, because what's different about seawater? What is different about seawater? It has salt. Very good. Okay. And that salt is not going to have the same water potential, the same movement as just regular water, because that salt is a concentration in your solution. So for your part C scoring, there's 4 points maximum. This is a long question. So you've got to talk about how water is going to exit the plant. And we already kind of know this. Water dehydrates you. That's why they tell you don't eat seawater. So you can kind of run with that. But it is a plant. So you've got to emphasize how this would be a plant cell as opposed to you. You've got to talk about osmosis, how these things are going to move across a membrane. You've got to talk about how water moves from solutions with higher water potential to lower water potential, and how that water can cause decreased crop production. So you've got to be very, very, very specific about how that salt is going to hurt that plant. Again, I know we haven't covered this in depth, so this one is kind of like, and I kind of hesitated to give C, but there is a 5-able on this weekend, I believe. I think it's Saturday or Sunday. So my encouragement to you guys is when I post the link to the slides, which I will be doing in just a moment, save this and try to work this problem on your own without looking at the scoring guide after watching the tonicity presentation, because you're going to be like, oh, I'll get this, and you're going to feel real smart. So that's the feeling we want y'all to have. Does anyone have any questions? Because I'm clicking and I don't have anything else. I think I'm done. Anyone have any questions? Anything I can help with? Especially people who have a test tomorrow? Junction proteins? Avery, your teacher has a lot of different stuff going on. So junction proteins are going to be, let's draw another picture. So junction proteins are real cool. These, if they're the ones I'm thinking of, are going to be when you have a couple of cells next to each other, and those cells have a protein that connects it to the other cells. So it's kind of like having a highway that connects a couple of states or a couple of countries. So that protein, all it's doing is connecting cell to cell. Any other questions I can help with? Does a single organelle do bulk transport, or is it the whole cell? That's going to be the cell membrane. The cell membrane is the part that's forming that, but then when it comes inside the cell, it becomes a vesicle. So that is technically a separate organelle when it's a vesicle, but that vesicle is made of cell membrane because it is membrane bound, it's phospholipids. Does that help, Omi-san? Awesome. Glucose is a yes, glucose is a monomer of a carbohydrate. Very good. It's polar and it's big. Good, good, good. These are good questions. Keep them rolling. Anyone have another one? I bet I can get it. I'm feeling tough tonight. What's degradation? I mean, to degrade something is to break it down. I haven't heard that in terms of a specific biology word. But when I say degradation, that means you're breaking something down. Does that make sense? Okay. Yeah, I don't know of any specific biology terms. But now I'm second guessing myself after Avery's asked about all these protein terms. So I'm like, man, maybe I haven't taught them enough. So any other questions? The ECM, the extracellular matrix. So the extracellular matrix is going to be, you got your cells. And there's always going to be cells connected to other cells. Fun fact, cells can't grow if they're not connected to other cells. If they do, that's cancer. They cannot grow unless they're connected to other cells. But we're going to have these little like gaps every here and there, and we're going to have fluid there. We're going to have different stuff. Most of the time, that extracellular matrix is going to be filled with proteins and communication molecules like hormones that are going to go and talk to other cells. Or it's going to be filled with stuff that you've eaten or drank that needs to get into your cells to nourish your body. Or it's even going to be some toxins that haven't quite made it all the way out of your body yet. Does that help? Awesome. Hit me. Come on, guys. Got 8 minutes. What do you need? If the solute concentration is high, is the water potential higher or lower? Okay, so let's check back here on this scoring guideline because it kind of lays it out for us. Okay. Concept of osmosis. The movement of water across a selectively permeable membrane from a solution with a lower solute concentration to a solution with a higher solute concentration. So we're moving from low to high here. So we're kind of going backwards. And it's moving from more positive, which is higher. So that's going to be lower solute concentration. So if the solute concentration is high, the water potential is going to be low. So if solute concentration is high, water potential is low. Think of it this way. Pure water is 0 water potential. That's how I kind of ground myself. So the more negative you get, the more solute you've got. Does that help, Audrey? Awesome. Very happy to help. What else y'all got? Are there differences in the functions of glycoproteins and glycolipids in cell membrane other than cell recognition? Not really. Nothing I can think of that you would need to know at this level. I can think of things when I was a junior in college, but nothing for AP Bio. Cool. What else you got? Hit me. Is that it? Did I do it? All right, y'all. It's 8:54 Central Time. It's about 12 minutes past my bedtime because I get up early. So last chance for any further questions, but otherwise I think we are good. Avery, good luck on your test tomorrow. Anyone else who has a test, best of luck to y'all. I hope this helped. I'll be back in a little while. It's all on the Fiberpool website. I can't keep track of my schedule, but it's all there. So if you enjoyed the presentation, make sure to post about it on the Twitter or the Instagram machine and come see me again. So thanks, guys. Glad you enjoyed it. Hope all your testing dreams come true. Bye-bye.